So class, we are going to, so these are the basic requirements you need for this practical on how to make a blood smear. Okay. So among the requirements, there are glass slides, lancets, cotton. So what we require for a prick, and then spirit for before pricking we need the spirit to clean our finger.
The surface of all the slides should be grease or oil free. Grease and oil free. Why? If there is any grease or oil while making the smear, you will find some part is black. No blood is there. Means like vacuole. So if it is there, if you get these vacuoles in your smear, that means these are not properly clean. So it should be clean at first. Then out of these two, one you will be using as spreader. One of the conditions of the spreader is the edge, particularly this edge or this edge or this edge and this edge. You have taken, but you don't know which edge you will be using. Okay, you have examined, you found this is the edge, fine. So you keep it like this while I will be using. So this edge only I will be using. You will find in most of the cases all the edges are same. So in that case there is no problem. It should be smooth. No cut or it should not be rough. Okay, no broken. It should not be broken. So this is one condition. And second, the first condition I said, the surface should be free. When it is cleaned by cotton, you place both the slides on a surface. Better you use a white sheet. From your notebook, you place it there on the surface of the table, then you keep both the slides. Everything cleaning done, as you have said, and thereafter never touch it at the center. Only thing, you can touch like this or you can hold like this. The spreader you will not be touching. This side you will not be touching. Spreader you will be holding like this. And this one you can, while using, only this way you will be holding. While you will be doing, you will be making the smear, you will be holding like this. This, it is on the surface. Come in. Okay. So this is from this side to, I will make smear from this side. So I will hold it like this. Okay. Then I will use the spreader from this end. And blood is, will demonstrate. So blood is at the back of this slide. Blood will be there, here. So I will bring the slide to the blood. I will bring the slide to the blood drop. Then when it is, the blood will be immediately spread along the edge, along the joint. Okay. When it is spreaded, blood is spreaded, then I will push it and the angle would be 45 degrees. I will tell in the procedure. So this is important. Yesterday what happened? Somebody pushed like this. See. The blood group is at the back of this. So somebody what did? This way, jerking movement. And when you will find, see the smear, it is also like that. Some part is thin, some part is thick, this way. So it should be neither slow nor so fast. Not slow. If you make it through slowly, then what will happen? The smear will be thick. If you make it fast, it will be thin. The blood will following will be following the edge of the. You are not pushing the blood. You are pushing the slide, but your blood will be following the slide. You remember this. Blood will be following the edge, following the junction of the slide, of both the slides. So its speed should be this. So before making smear, you practice this at a time. Understood? So before making the smear, you practice like this. Okay, then you will find it is okay. Make it. Okay. Go.
So the stain we are using, it's called Leishman stain. Okay, the spelling is written here. It is a group of stain that falls under a name called Romanowski group of stain. Now, what is Romanowski group of stain? These are some special stains
group
that
of stain.
Two different staining material that stains both the acidic and basic part of a cell. Acidic part like nucleus. Why nucleus is the acidic part? Because nucleus has nucleic acid. Deoxyribonucleic acid means DNA. So that is the acidic part. So for staining the acidic part, we need a basic dye. For example, in this case, we are using Leishman stain. Leishman stain has three main components. What are they? First part is eucin, second is methylene blue, and the next, important part, started staining, it's acetone-free methyl alcohol. Now, what is the role of eucin and what is the role of methylene blue? Eucin, you know, EOSIN, just like the name from the cell called eosinophils. Eucin is used to stain the eosinophils. Eucin is actually a acidic dye. So what will it stain? The basic part. Okay. Eosinophils is a type of WBC which has basic granules. Okay. So it will be stained by the acidic part of Leishman stain. Got it? Now it has also another stain that is called methylene blue. Methylene blue is a basic dye. So it will stain the acidic part like nucleus. So the nucleus of WBC will be stained by new methylene blue, and you will see the bluish tinge is there. Okay, and the granules of eosinophil will be stained by eucin, and eucin also stains hemoglobin. Okay, and the granules of basophils will be stained by. Basophil has basic now, so it will be stained by some basic stain. Okay, eosinophil is stained by eucin. Basophil will be, the granules of basophil will be stained by this methylene blue. So, and the next component is acetone-free methyl alcohol.
I think she is having some confusion, isn't it? Hey,
but-
that. How can? I just observed your face.
This basic diet.
Yeah,
So it will stain the acidic.
acidic part, like nucleus. Nucleus is acidic because of
It's acidic.
eosinophil का जो content है वो basic है. ठीक है? The granules of eosinophil are basic, so it will be stained by the acidic dye, which is eucin.
Histones are there. Lot of histones are there. Histones are basic.
The granules of what I just told, basophil. So see, eosinophil will be stained by eucin, and the basophil, the granules of basophil will be stained by the opposite one, that is methylene blue.
So the granules of acidic will be acidic.
Then it should be acidic. Yes, acidic. Now clear? Okay. And there is another content, that is acetone-free methyl alcohol. The function of methyl alcohol here is to settle that content of the cell, that is called fixation. When you are making a smear, it should be fixated to the glass slide, or else when you are using the stain and when you are using that
It doesn't wash away.
When you are using the tap water, it will be washed away. So for fixation of the blood smear with the slide, we need that methyl alcohol. So methyl alcohol helps in fixation. Okay. And why it is acetone free? Acetone actually causes shrinkage of the RBC. Okay, when if the methyl alcohol has some acetone in it, the RBC size will be shrinked. So to prevent that shrinkage, we use acetone-free methyl alcohol. And methyl alcohol actually preserves the proteins and other particles that are present in the smear as it is in its chemical and biological form, so that it doesn't change.
Done?
Yes, sir.
Okay. So now we will go to procedure. What is that procedure? Actually, this will come later. This is the initial part only. So you see, now this slide is perpendicular. What is the angle? 90 degree. So I want to make it half. It's almost half. So this is how much? 45 degree. So this is the angle by which this angle should be at 90 degree angle with this slide. So then only maintaining this angle, you will be pushing the slide. Not like this, not like this, but like this. You should not, while you moving, it should not be lifted.
After we have done with the pricking, by wiping the middle part of the tip of the finger, the palm of the finger, using a spirit and then drying it up, we are going to prick it, and after pricking we are going to wipe the first drop of blood, and then you are going to place a small drop of blood, not much big, not much small also, very tiny, but medium sized drop of blood at the center of the glass slide where you will make the smear. Center and 1 to 2 centimeter away from, just see what sir is doing, at the
One
center, means midline.
One side, but at the midline.
1 to 2 centimeter from the edge, not at the very edge. 1 to 2 centimeter from the edge, you are going to place your finger and make that blood drop. Okay?
Break. And how much, after placing the drop, making this smear, how much time you will take? 3 minutes, 4 minutes, 5 minutes? Why? So before blood gets clotted, so before that I have to finish this. So time is also important.
So after you have placed the drop of blood at the center 1 to 2 centimeter away from the edge, now you are going to use your spreader. Spreader means the glass slide which is having the blunted and on the smooth edge, not rough edge. Okay, if you are using a rough edge, the tail part of that smear will be like this. Okay, like zigzag line. Okay, so to have the tongue-shaped perfect smear, you will be using a spreader that's edge is blunt or smooth. So see, it has been kept at an angle of 45 degree, not more, not less. What will happen if the angle is?
See, it is placed like this. No, it should be placed like this. In the picture, it is a little bit curved, no? So
Just like this, at an angle of 45 degrees. Okay, you are going to use that spreader. Starting from the other end, you are going to bring it back and touch the blood drop. The blood drop will then spread at behind the blunt edge of the spreader. It will spread, and after it is spread, then you are going to push it forward. Okay, at the same angle, at 45 degree angle only, you are going to push it forward. Not so fast, not so slow. At a steady speed. Okay, not like that, not like that also, and not like stopping in between. Like smooth, and then I'm going to push it, and you will see there is formation of that perfect smear. Now, if you are making the angle more than 45 now, then what will happen? Most of the blood will be staying at the first part only. It will be making a thick smear. And if you are making the angle less than 45, like 30 degree, 20 degree, like this, very slanting, then the smear will be very thin. So to make a perfect single cell layer smear, tongue-shaped smear, you are going to make the angle of the spreader with the slide at 45 degree angle. Got it? See, what are the differences between an ideal smear and the smear that is not ideal? As ideal smear, see, it has a tongue-shaped nature, and see there can be vacuums. See, in the third slide, the film is too thick. Either he has taken more blood, or the angle was not too perfect. That means the angle was more than 45 degree. And see, I told you now, if the edge of the spreader is not smooth or blunt, then you will see this kind of smear, that is at number 4. Okay, so to avoid all this and to make the smear as ideal as possible, we are going to do the precautions what we have just discussed. Okay.
Why there is vacuole there? In the second slide, we can see there are two vacuoles. What is the reason of this?
So these are the procedure that have been taken from your book only, AKJ.
You.
Okay, one of you will come here and tell the points.
Read out slowly, not so fast. Go there.
What is your name? Okay.
So this is the procedure for preparation of a blood smear. The first step is take 4 or 5 clean, grease-free glass slides with smooth edges, 3 to 4 to be covered with a blood film and 1 to be used as a spreader. Step 2. Lay 3 or 4 glass slides on the table, prick the finger, touch the bleeding point lightly in the center line about 1 or 2 centimeters from one end of the slide to obtain a small drop of blood on each slide. Now step 3. Place the narrow edge of the spreader on the surface of slide just in front of the blood drop at an angle of 45 degrees as we have been shown in the figure. Draw the spreader backwards so as to touch the blood drop and hold there till the blood runs along the full width of the spreader at the line of touch. Then step 4. The spreader is then moved slowly and smoothly to the other end of the slide, maintaining an angle of 45 degrees. And the last step, allow the film to dry here. Repeat the procedure with the other slides. Now, these are the procedures for fixing and staining of the blood smear. First, fixing the blood smear. Step 1. Place the blood smears across 2 of the parallel supports on the glass rods so that the slides may be horizontal. Now step 2, pour.
Wait. How many drops? You have to pour. How many drops? How many? Remember? 8 to 10 drops of the surface of the smear.
Next.
Continue.
Step 2, pour 8 to 12 drops of Leishman stain on each slide, just enough to cover blood smear. Note the time. Leave it for about 2 minutes, which is called the fixation time.
2 minutes.
Now the procedure for staining of blood smear. Step 1. At the end of 2 minutes, add an equal number of drops of buffered water of pH 6.8 over the stain. Take care that the water is not spilled over.
Okay? Same amount of buffer solution. If buffer solution is not there, we will be using distilled water, but double the amount of buffer.
Step 2. Mix the stain and water evenly by gently blowing air intermittently with the help of a dropper. Step 3. If the dilution of the stain is correct, the fluid will be covered by a thin greenish scum. Leave the stain to act for 10 minutes called the staining time. Now, at the end of 10 minutes, pour off the stain. Hold the slide in a slanting position below the tap and the water is allowed to flow over the smear. Next step, wash the slide up to 2 minutes with tap water gently and thoroughly till the film gets a pinkish tint. Make sure that number 1, none of the greenish scum settles on the surface of the blood film. Number 2, water stream should not strike the blood smear directly or else it may be washed off.
After that, what?
Wipe clean the back of the slide and set it upright to dry.
Next.
Okay. So,
Okay.
give me the.
Now you go.
Okay.
You will tell. Now you will tell the procedure.
So you have seen the procedures, no? So the first 2 minutes is for fixation time, and the next 10 minutes is for staging time. So actually the fixation time, I told you, na, something in the Leishman staging helps in fixation. What is that? Acetone-free methyl alcohol. That helps in the fixation of the smear. So the first 2 minutes is for fixation. Okay.
Where you will keep the slides before you apply stain. Where you will keep?
In that parallel rods, see, na? There are parallel rods. In between the parallel rods, you will.
They are horizontally placed. You keep there like this.
And at the same time transports metabolic waste away from the same cells. Vertebrate blood is composed of blood cells suspended in plasma. Blood cells mainly consist of red blood cells or erythrocytes or RBCs, white blood cells or leukocytes or WBCs, and platelet cells. RBCs are the most abundant blood cells. They impart a red color to the blood. In this experiment, we will be covering the procedure on how to prepare a thin blood smear and stain it with an appropriate stain for the morphological and quantitative study of the various cellular elements of blood. For this experiment, we will need the following: blood sample, glass slides, Leishman stain or Wright's blood stain or GM's stain, distilled water, droppers, petri dish or coupling jar, compound light microscope, immersion oil, cotton and rubbing alcohol, blood lancet or a pricking device. Take 2 glass slides and clean it with 90% alcohol. Disinfect a finger with alcohol and prick with a sterile lancet or a pricking device. Place a tiny droplet of blood in the center of one corner of one of the glass slides. Using a second pre-cleaned glass slide, touch the drop of blood while inclining the slide at a 45-degree angle. Now gently and briskly move the inclined slide towards the other end of the slide containing the blood drop in order to obtain a smear. A properly smeared blood appears roughly tongue-shaped as seen here. Allow the smear to air dry for a minute or so. Place the slide in a petri dish with the blood smear facing up. Add several drops of the Leishman stain to the blood smear and cover the petri dish. Alternatively, you may fill a coffin jar with Leishman stain and immerse the slide in stain. Stain for about 1 minute and then add twice the volume of distilled water to the stain. Allow the water and the stain to properly mix and keep it aside for about 10 minutes. Drain the slide and wash with distilled water. Proper staining results in a blood smear that is rose pink in color. Air dry the slide while keeping it in an inclined position. The slide is now ready for observation under the microscope. Observe and study the various blood cells in the 40x and 100x objective. For 100x, place a drop of immersion oil on the slide. Gently lower the 100x objective until it touches the oil surface. Here on, use a fine adjustment knob for focusing. The following microphotographs show the various cellular components of human blood.
See, the basal nucleus cannot be distinguished. Nucleus, outline of nucleus cannot, and at the surface of the nucleus there are a lot of granules. So this is
ওই নিশা স্পিরিটটা দিত স্পিরিট বালা একটু স্পিরিট বালা দে
আর একটু উপরে ওঠা, এটা পড়বে। হ্যাঁ, কারেক্ট। পিছিয়ে আন। নাইস। আইসে আইসে আই পড়ছি।
Yeah.